Okay, so if you enjoyed today's session, make sure that you also join our upcoming Genetic Essential Training series. And hopefully you have registered to all of our sessions, but if not, feel free to scan the barcode here or register at the bit.ly/nala-get. Next. Okay, so with all of our amazing products that I shared just now, I think we are also lucky because we have exclusive offers just for today's webinar attendees. That is a buy one, get one free and a 30% off for selected tests. So I'll explain more on the next slide. So the buy one, get one free is for our genetic testing panels, including RX Ready, that is for personalizing medication, Mammo Ready for breast cancer, Cardiac Ready for cardiovascular health, Nutri Ready, and Life Ready for your diet and wellness. So you can purchase these tests individually or bundled, and you will save more with bundles. So if you are interested, this is a very limited offer. Please contact the WhatsApp on the screen now, or if you want to think first, just take a screenshot or take a picture of this screen right now. And we also have an even more amazing offer that is a special discount up to 30% on two tests that are related to today's topic. So the first one is our hereditary cancer panels, which includes colorectal, pancreatic, gynecological, prostate, skin cancer, and more. And the second one is our somatic tumor panel, which is for non-hereditary cancers, including lung, colorectal, endometrial, thyroid cancers. And if you are still not sure what these tests are, don't worry, because during today's session, our speakers will explain all of them to you. But please remember that the special discount is only for the webinar attendees today. Okay, next slide. Okay, now let's welcome our first speaker, Dr. Natalia Zanziaras, Master of Science. She graduated from University of Samratulangi for her medical doctor degree in 2010 and her Master of Science in Genomic Medicine from Imperial College London in 2021. And today, she is serving as our amazing genetic counselor in NalaGenetics and also a lecturer at Medical School of Satya Wachana Christian University. Today, Dr. Zensi will be sharing about a new era in proactive cancer screening. Just a reminder to our attendees, if you have any questions during the talk, feel free to type them in the chat using the format name_institution_your question. Okay, Dr. Zensi, the screen is yours.
Thank you, Dr. Salma. Good morning, everyone. I need help for the slide, please. Okay, thank you. Good morning again, everyone. Today, I'm representing NalaGenetics. We are on the cusp of a significant shift in how we approach cancer. And today, I want to talk about the new era in proactive cancer screening. Next slide. Thank you. We've made incredible strides in treating cancer from researchers, scientists, clinicians, from chemotherapy and surgery to advanced radiotherapy, immunotherapy, and targeted therapies. Our arsenal against established cancers is more sophisticated than ever before. So now we can precisely target tumors, harness the body's own immune system, and develop personalized treatment plans. But this leads to critical questions. What if we could apply that same level of sophistication before the cancer even develops? Are we truly doing enough to predict and prevent cancer in our patients and indeed in ourselves? Next slide. Today, at this session, we will explore the path to proactive screening. We'll start by examining the limitations of our traditional population-based screening methods, and we'll delve into the genomic shift, understanding inherited risk beyond just family history. Following that, we'll discuss the clinical tools that translate this data into actionable insights. And finally, we will look at how to integrate this into practice with a compelling case study. Next slide. So let's begin by looking at where we stand today, the current landscape of cancer screening. Can look at the table, our current wait and see model, which while effective to a degree has inherent limitations. It is largely based on a one-size-fits-all approach, relying on age-based guidelines for screening. For instance, mammograms every year for women 45 to 54-year-old, or colonoscopies every 3 to 10 years based on the risk. And the fundamental issue here is that this model is based on age, not based on individual risk. This leads to what we call interval cancers. That is cancer that tragically develop and progress between scheduled screenings, often when they are already more advanced and harder to treat. Next slide. Furthermore, traditional screening is often one organ at a time, like a mammogram for breast, a colonoscopy for colon. This excludes other cancer types that can be cost inefficient. It also often requires experienced personnel for decision-making and can sometimes be invasive. We imagine a world where one test rules them all, a single modality capable of detecting multiple cancer types. This is the promise of multi-cancer early detection, or MCED. It is cost-efficient, non-invasive. It can integrate AI into decision-making and holds the potential for truly universal cancer screening. This is the future we are building. Next slide. And now, let's talk about the paradigm shift, moving from population risk to individual risk. For a long time, family history has been our primary tool for assessing hereditary cancer risk. And while it is crucial, it is far from perfect. Factors like small families, unknown parentage, male carriers of female-centric cancer genes like BRCA, or even de novo mutations. De novo mutation is a new mutation not inherited from parents. All of these can obscure a patient's true genetic risk. In fact, clinical studies consistently show that nearly half of the patients with pathogenic germline variant are missed when we rely solely on the traditional family history-based testing criteria. So we need more comprehensive approach. Next slide. To understand this, let's briefly touch on the genomic basis of hereditary cancer. It is often explained by a quite popular hypothesis, that is Knudsen's two-hit hypothesis. We are talking about tumor suppressor genes. Tumor suppressor genes act like a brake pedal for cell growth and DNA repair, like BRCA1 or TP53. In hereditary cancer, if an individual is born with one faulty brake, this is the first hit. So a germline variant present in every cell. We only need one additional somatic event, that is the second hit, to damage the other functioning brake in a specific cell. So with both copies lost, cellular control fails, dramatically increasing the probability of cancer development, and it's often at an earlier age. This leads to a spectrum of risk. High penetrance genes like TP53 or BRCA1 confer a very high lifetime risk, often 40 to 80% for specific cancers. These have traditionally been the focus of limited genetic testing. However, now we recognize the importance of moderate penetrant genes such as ATM, SHAP2, PALB2. These genes significantly increase lifetime risk. around 20 to 40%, and are often missed by narrow criteria-based testing. Yet they represent a major opportunity for personalized screening and prevention. Next slide. Okay, so what is the key biomarker enabling this new era? It's DNA methylation. Think of it as an on or off switch for genes. There's not a change in the DNA sequence in, itself. So imagine your DNA is like a giant instruction manual for building and running your body. So this manual has thousands of recepts like genes, called genes, which tell your cells how to make proteins, how to do their jobs, and how to grow properly. Now think of each genes as having its own volume control knob. So DNA methylation is like a tiny chemical text, specifically a small molecule called methyl group that can attach to certain spots on your DNA, usually in the front of a gene. So what does this volume control do? It could turn the volume down or off. So when these metal tags attach to a gene, they can make it harder for the cell to read the genes. So it's like turning the knob down or even completely off. This means the cell makes less of the protein that the gene is supposed to produce, or sometimes none at all. Or it could keep the volume up or on. So if there are no methyl tags or very few, this gene's volume stays up and the cell can read it easily and makes lots of its protein. Why is this important? Every cell in your body has the exact same DNA instruction manual. But a skin cell, for instance, a skin cell is very different from a brain cell, right? That's because DNA methylation, along with other volume controls, helps cells decide which genes to turn on and which to turn off. It is how a skin cell knows to act like a skin cell and not try to grow into a brain cell. Now here is where it gets really interesting, especially to understanding cancer. In healthy cell, DNA methylation patterns are very organized and predictable, but in cancer cell, these patterns often go haywire. Like in tumor suppressor genes get silenced. So imagine you have the stop genes that are supposed to prevent cells from growing out of control. In cancer, metal tags often stick to these stop genes, turning their volume down or off. So this is like removing the brakes from a car. So the cell can grow widely. Or it could growth genes get turned on. So sometimes the genes that promote cell growth, that is called oncogenes, get their volume turned up, and when there shouldn't be. Scientists now can look for these abnormal DNA methylation patterns in tiny bits of DNA that cancer cells shed into the blood. Cancer creates unique and highly stable methylation patterns. So it is like finding a unique fingerprint of cancer in the blood sample as circulating free DNA or circulating tumor DNA, even before a tumor become large enough to see on a scan. And this is why it is such a promising tool for multi-cancer early detection. The critical advantage of DNA methylation is its ability to accurately predict the cancer source tissue, for instance, pancreas, colon, or lung. So this fills a crucial gap between assessing risk through germline testing and confirming a diagnosis through imaging. It allows for a faster and more guided clinical workup. So this was powerfully demonstrated in a recent Pathfinder study. You can look it up. It was published in the Lancet in 2023, where the MCED test not only detected dozens of early stage cancers, but also predicted the tissue of origin with high accuracy, leading to confirmed diagnosis in less than 3 months for most cases. Very interesting. Next slide. So given to this powerful new tool, who should be considered for proactive screening? We can think of this in two tiers. The first one includes those with clear indications, so a significant personal or family history of cancer. So this is our traditional group. But the new era brings us the second tier, expanding the indication significantly. So this tier includes healthcare professional with occupational exposure, patients with a limited or unknown family history, individuals with high anxiety about cancer risk seeking definitive information, and frankly, anyone seeking a comprehensive proactive baseline for their long-term health plan. This is about empowering individuals with knowledge about their unique risk. Next slide. So how do we integrate this powerful genomic approach into the clinical practice or into the practice? It can be broken down into 4 key steps: identify, counsel, test, and manage. The first one is identify. We broaden our clinical lens. We move beyond just strong family history. So we consider patients with limited or unknown family history, those with professionals or environmental risk factors, or simply anyone desiring proactive health information. The second is counsel. We set the stage for an informed decision, conduct pre-test counseling, discussing potential outcomes, positive, negatives, or a variant of uncertain significance or VUS, explain the benefits like personalized screening and of course the limitations, and understanding that not all cancer is hereditary, and of course most of the cancers are somatic. And third is the test. We utilize validated genetic analysis, order a comprehensive multi-gene panel from a certified and accredited laboratory, ensure it analyzes a wide range of clinically relevant actionable genes associated with hereditary cancer risk. The process is simple, typically requiring only saliva or blood sample. And finally, the last one is manage, translate that data into clinical action plan. So for a positive result, we develop a personalized surveillance and risk reduction plan based on established guidelines like those from NCCN. This could mean earlier mammograms, adding breast MRIs, or more frequent colonoscopies. For a negative result, we provide reassurance and recommend adherence to standard population-based screening. This result is also clinically valuable. So always leverage post-test genetic counseling to help patients truly understand and act on their results. Next slide. Okay, we come to the slides of case study. To illustrate the real-world impact of this approach, let's look at the case study. Let's meet Bapak X, Mr. X, a 58-year-old executive from South Jakarta. He's a non-smoker with a relatively active lifestyle. He went to his doctor with a 3-month history of troubling but non-specific symptoms such as persistent deep fatigue disproportionate to his workload, vague, intermittent upper abdominal discomfort, and unintentional weight loss. Initial workup, including complete blood count, metabolic panel, and abdominal ultrasound, written inconclusive result. So Mr. X remained anxious and still symptomatic. At this point, a liquid biopsy using DNA methylation profiling was performed to screen for an active cancer signal in his blood. The test was positive. So this crucial result guided a targeted MRI, which confirmed a small stage 2 pancreatic tumor. The early and precise diagnosis made Mr. X a candidate for curative intent surgery, dramatically improving his prognosis. In this case, perfectly exemplifies how proactive screening can change life. Next slide. well, we come to the end of the slide. Okay, thank you for your time and attention today. So we believe this new era in proactive cancer screening, driven by genomic and epigenetic insights, holds immense promise for transforming patient care and moving us closer to a future where cancer is detected earlier and managed more effectively and ultimately prevented. And I'm happy to take any questions you may have. You can, if you are, for Indonesian, if you are more comfortable to speak in Bahasa Indonesia, it's fine too. Thank you. Back to you, Dr. Salma.
Thank you so much, Dr. Zenzi, for the very insightful presentation. So let's open for Q&A. If anyone has any questions, you can put it on the chat box or raise hand. Kalau ada yang ingin bertanya dengan bahasa Indonesia juga dipersilahkan. Okay, we have a question from Dr. Raisa here. Thank you, Dr. Zenzi, for the explanation. I'd like to ask whether this technology for DNA methylation detection is ready to be applied in Indonesia. Thank you, Doc.
Good morning. Thank you, Raisa, for the questions. Yes, yes, you can. Actually, this session is for to introduce this new era promising of the preventive and diagnosis. That's why you need to stay until the end of the of the sessions, and there will be more interesting stuff going on after this. And yes, you can contact NalaGenetics for further informations and for the operational information how to do the test. Thank you.
Thank you so much, Dr. Zenzi. Hopefully that answers to Dr. Raisa. We have another question here from Dr. Taufik. Okay, one moment. Okay. From Dr. Taufik here regarding false negatives, Doc. Are there known tumor characteristics such as low shedding rates, specific mutational profiles, or certain histological subtypes that are likely missed by the current ctDNA platforms? And how should we counsel a high-risk patient who receive a negative MCED result?
Yeah, thank you. This is Dr. Taufik. Dr. Taufik, after this, we will have another session. I think Mr. Dr. Suarez will answer your questions after the sessions. So I think it's on his slide and so on, or we can discuss it after the sessions, including the limitations of the technologies and so on. It will be discussed after my sessions, Dr. Salma.
Okay, noted, Doc.
Yeah.
Okay, so to Dr. Taufik and Dr. Lona, please stay until the end of the session. I think the discussion, the Q&A will be more comprehensive, yeah, Doc, after
Yeah.
Dr. Suarez's session. Okay. This is a question regarding genetic counseling, Doc. I think this is perfect for you. This is from Dr. Ziske Maritska. Dr. Zwensi, thank you for sharing this morning. I'd like to ask what are the things that you would like to emphasize in the pre-test and post-test genetic counseling? How does it differ from the current genomic cancer risk? Do you still use some checklist tools to assess the risk?
Yes, that's a very, very good question. For me, my best is the patients know that what kind of the technology that we are using, what's the difference of the technology that they've done before, and of course we need to discuss the limitations and what is the expectations, because patients come to us with different background and they have different expectations. Some of them they just want to do like general checkup without family history of hereditary cancers, and some of them come to with a lot of anxiety because of the family history the family hereditary cancer that are quite strong in the first or second tier of the family. So I have different approach, but the main my main goal is the patients know what to expect and know the limitations, and we as a counselor, we know what is their main reason for doing the test. because this new era give a broadened indication, right? So not just patients with family history. Thank you. And I think we can discuss more in onboarding, because this session is like the timing is quite short for genetic counseling discussion. Thank you.
Yes, thank you so much, Dr. Zwansi. Yeah, so if I can summarize, it would be to make sure that we facilitate the patient to understand what is to be expected from the test and what are the limitations of the test, and for us to also understand the main reason for the patient doing the test. Okay. There's a follow-up question from Dr. Ziske.
Mhm.
When it comes to current genomic cancer risk test, the limitations are that the genes being tested are those of
Mhm.
low and moderate penetrance. What
Mhm.
are the limitations of this new test, the ctDNA?
So actually, we've got a different kind of test. We test for high penetrant genes as well. That's why it depends on what's the main reason or main indications of each patient, right? So and for these DNA methylations, you have to look at the technology because it's looking at the epigenetic. It's not it doesn't sequence the whole. This is not we are not talking about whole genome sequencing. So they look at the epigenetic tier of DNA. So this is something that patients need to, because patients normally they will come like, what is the difference? What is the difference? So what could make the difference of this? So we need to explain about the ctDNA as well and so on. So we need to explain about the a little bit of technology, the difference between the scale of DNA and the scale of RNA expressions, and now we are talking about DNA methylation in epigenetics. Thank you.
Okay, thank you, Dr. Zansi. So maybe for Dr. Ziske's question is similar to Dr. Lona's question, which is regarding the limitation of this technology. So we will learn more from Dr. Swaraj
Mhm.
for the second session on the technology itself. And maybe we can come back to Dr. Zwensi later on how to maybe deliver the knowledge that we gain from Dr. Swaraj, how can we deliver that to the patient, maybe in more layman terms? So, to Dr. Ziske and Dr. Lana, please stay tuned to the end of the session. And also, I think just to add on, for Dr. Ziske, who are saying the limitation of current genomic cancer risk tests are testing low and moderate penetrance genes. Actually, for our own in-house test, that is Mamoredi, Dr. Zanti, we also
Mhm.
test for high penetrance genes, including BRCA1, BRCA2, and PLB2. So if Dr. Ziske is maybe curious on Mammo-Ready test, which also tests on high penetrance gene, feel free to later discuss with NalaGenetics team. We're looking forward, I think, to discuss more with you. Okay. Any other questions? Let's wait for a few seconds, Doc. Okay, there's a question from Dr. Gavian Hua. Are there specific guidelines, Doc, in deciding when to order DNA methylation tests versus other diagnostic tests? So on the indication.
Yeah. Until now, not yet. Yeah. So we just we look at the some of the papers, and we have our in-house indications. And I think from strength will Mr. Swaraj, Dr. Swaraj will explain this too.
Okay, noted, Doc. Thank
Yeah.
you. So regarding specific guidelines from medical associations, currently there is still none yet, Doc. Yeah.
Mm-mm.
Okay,
Yeah.
but we can confer from uh, published studies and decide on our internal uh, guideline of our indication. Okay, any other questions from the attendees? Let's wait a few seconds. Okay, if there is no more question for Dr. Zwensi, Thank you so much, Dr. Zenzi, for sharing your insights and knowledge. Please don't go away, Doc. We still have the second session, and
No worries,
uh,
no worries. Yeah,
we'll
I
invite
will.
you back.
Sorry, Dr. Sam, I will add about the guidelines. I forgot that. I mean, I think in NCCN they didn't mention specifically about DNA methylation, but there are some guidelines about genetic screening or something sort of like that, like about the preventions and testing the high penetrant genes, testing the low to moderate penetrant genes, and so on. You can look it up at NCCN, but I'm not very sure about talking specifically about DNA methylations. That's it.
Yeah. Okay. Noted, Doc. Thank you so much for the clarification. Okay, Doc, I will invite you back for the second discussion and Q&A session. So now let's continue to the next session. Oh, but before that, just a reminder, we have a special promo, buy one, get one free, and a 30% off for selected tests. So next slide. Our buy one, get one free is for our pharmacogenomics test, that is RX Ready, Mammo Ready, that is our breast cancer risk prediction test, Cardiac Ready, our cardiovascular disease risk prediction test, Nutri Ready and Life Ready for those of you who want to personalize your diet, your fitness, your skin health, your lifestyle in general. Um, of course, you can buy this in individual tests, but you will save more with bundles. So I would really recommend to get the bundles. You can buy this for yourself, for your patient, or for your loved ones as well. So if you buy one for yourself, you can share the free one maybe for your loved one. And next slide. We also have a special 30% discount. So this one is for the test that we are explaining in this session. So we have the hereditary cancer panel and the somatic tumor panel. The price starts from 4 million IDR or 340 SGD. So this is a very good deal, and it's only for the webinar attendees today. If you have, if you are interested or you have any other questions, feel free to contact the WhatsApp that is on the screen right now. Okay, so uh, now up next we have a speaker who is going to take us into more on the technology and the molecular details of ctDNA analysis. So please welcome Dr. Swaraj Basu, who will be presenting uh, Decoding Early Signals, the Science and Technology of ctDNA Methylation Analysis. But let me first read his bio. Dr. Swaraj Basu completed his Bachelor of Science in Botany Zoology Chemistry from University of Allahabad in 2005 and Master of Science in Bioinformatics and Biotechnology from SRM University in Chennai. He was also a research fellow at the National Institute of Plant Genome Research in New Delhi, and he completed his PhD in bioinformatics from Stazione Zoologica Anton Dorn in Italy, and his postdoc in 2019 from the University of Gothenburg in Sweden. Currently, he is serving as the principal bioinformatics engineer at Strand Life Sciences. Okay. So without further ado, Dr. Suarez, the floor is yours.
Thank you. Thank you for the kind introduction, and thank you, Natalia, for this quite insightful presentation before. Uh, uh, very, very well put points. Before I start, just wanted to check, is my audio okay for everyone?
Yes, it's okay on my side.
Perfect. So I'll share my screen and get started. My screen is visible now.
Yes,
Perfect. Thank you.
it's visible now.
So I'll take everyone through a journey which we at Strand Life Sciences also feel quite proud of, but we also think it's a continuous work in progress. We'll keep coming up with better science and more updates. The idea of it starts, let me go back a bit to the historical context. And the historical context is that we have a very broad timeline of these various therapies or drugs which are used in oncology, starting with imatinib quite early for myelomas, and then we have crizotinib somewhat around in 2010 and later for non-small cell lung cancer, and more recent is sotorasib, which is quite interesting because a lot of AI modeling was used for the first time in development of a drug. So while we were having this evolution of these drugs for oncology, in parallel, we also had a big change, or I might say revolution, in the way we detect and diagnose disease. And that detection and diagnosis came from the fact that in the early 2000s, we had microarrays. From there, we had RNA arrays. We got the human genome draft. We got NGS, which reduced the sequencing cost. We got 1,000 genomes, whole exomes, so many things. And somewhat around 2015, we also started getting NGS-based methyl-seq assays. I'll talk more about methyl-seq and why it's interesting. Of course, Google launched its deep learning biomarker detection tool, Deep Variant, getting adapted quite often. Nowadays, we have single cell and spatial transcriptomics. And as we speak, early 2020s, sotorasib was one of the first AI-driven drug discovery where the binding site was actively understood based on various computational models. So why I'm showing this? Because I think our view into our past tells us a lot about what we are going to achieve in our present and the direction for our future. This evolution shows that therapy and diagnosis or screening are running together, both pacing ahead in the AI landscape. More and more things are coming up, and probably I wouldn't be surprised if what we are going to achieve in the next 10 years is going to outpace what we have done in the last 50 to 80 years. So this brings me to a question that when we go for any kind of well-known cancer diagnostic tests, like a mammography with 70 to 90% sensitivity, lung CT scan, 60 to 90% sensitivity, again, different sources. We have pap smear, HPV test, colonoscopy, upper endoscopy, liver ultrasounds, transvaginal ultrasounds. Each of these takes a day, half a day at the clinic, appointments. Of course, it is accompanied by guidance from a clinician, some early symptoms, probably quite often not when it is detected, the stage of detection could be late. Can we augment all of these fantastic existing technology with a single test? And that single test is blood sample, can be taken at any point in the life duration of an individual, like recommended above 50 individuals above 50 years of age. Can it lead to early intervention? From one blood sample, can we try and detect with a substantial sensitivity and specificity presence of tumor DNA from multiple cancer types? So one test for all kind of situation. And can this be routinely introduced in our clinics as a screening protocol? And this is where Strand Cancer Spot comes in. What we have shown is close to 79% sensitivity just from a blood test, 10 to 20 ml of blood, early stages of cancer, stages 1 and 2, at 97% specificity. Now, since I talked about augmenting existing protocols with Cancer Spot, which is a multi-cancer early detection test, what do we use in Cancer Spot? We use methylation. In methylation, what we have seen repeatedly and what you are seeing on my screen right now is sequencing data. Sequencing is nothing but getting all the bases which DNA has. That DNA can come from blood, tissue, any source. And here you can see that this is a healthy individual. This is a certain position in the genome. And in this particular position, this is a CpG site. And it is unmethylated. These are all Ts. We don't get to see any methylation. This is how methylation experiments work. Where there is no methylation, sequencing data shows us Ts. And this is a colorectal cancer patient. And here at the same spot, it always shows C. Now, this is quite like a mutation, but it's far, far more widespread. It's almost everywhere in the genome if we know what are the positions which need to be captured. And where this data is coming from, this data is coming from sequencer. What was fed in the sequencer, DNA from a healthy individual and a cancer patient. So basically what I'm showing here is I can keep repeating this measurement multiple times in these two persons. Every time I'll get lots of T's, every time I'll get lots of C's here. And this forms the crux or the basis of our technology, the methylation, early cancer detection with methylation. Now, since we are talking methylation, let's have a quick revision on what methylation is. There are certain enzymes in our body. They will DNA methyltransferases. They remove the H group from cytosine and replace it with a CH3 group. Why do they do that? Classically, we knew that DNA is wrapped around histones. And when this CH3 group is added, it makes more tighter coiling and it makes DNA less accessible. While there is no methylation, DNA is more accessible. So methylation is kind of an epigenetic regulatory mechanism. Recent papers are also telling us a different story. They are saying that not only methylation can induce tighter conformations, in certain cases, methylation can also induce loose conformations around DNA, around histone proteins. So again, as always, biology being a science of exceptions, we can see both kinds of behavior. That methylation closely confirms DNA. Sometimes methylation also loosely confirms DNA. But what is well established is methylation indeed guides DNA availability for other processes, transcription, enhancers, and other things. So that's why it is very crucial that a phenomenon like this, which constantly switches on and off large regions of our DNA, depending on tissue specificity, depending on what kind of underlying cells they are, do they do something different in cancer cells? And the answer is yes, clearly yes. And this brings us to the point of non-invasive multi-cancer early detection. As I put forward, can we put a non-invasive multi-cancer early detection test as an augmentation to the existing clinical protocols? A study has clearly shown that cure fraction, which is, can a patient reach a stage in cure where the patient's mortality is no less than the average mortality of all other healthy people of that age group? Can a substantial fraction of patients be cured if cancer is detected early? And the evidence is overwhelming across multiple cancer types. Stage 1 and 2 detection often leads to twice the chance of cure for multiple cancer types compared to detection in stage 3 and 4. Even with cancers like ovarian or lung, where early stage detection also has low success rate, early stage detection still improves the cure fraction far more compared to late stage. And that brings us to non-invasive cancer detection tests.
Swaraj,
Yeah.
sorry to cut you off. Would you mind collapsing the Zoom video on
Sorry.
the right
Yeah.
side so that we can see your full slide? Yes, thank you so much.
Sorry. I'm so sorry. Yeah, thank you.
No worries. Thank you.
So this brings us to the question that what do we do out of the blood? Now we know that we are measuring methylation. Now we know that we are talking about tumor uninformed approach. We don't know what to biopsy, where to biopsy. We don't want to spend a day at the clinic with the clinician so early. We want to take a rough screening estimate and then take a clinical direction out of it. The answer is the various body fluid sources. So what we have seen is tears, breath, aspirate fluid, apocrine sweat, urine, and primarily blood. They all contain small fragments of the DNA secreted by our cells. Now, in blood, these can be circulating tumor cells if a person has tumor, proteins, cell-free DNA, which is what we are kind of focusing on, microRNAs, extracellular vesicles, and metabolites and the idea is that if there is an early stage tumor and if those cells are shedding DNA in the bloodstream, can we capture that DNA and can we do something out of it, like measure the methylation signal of the DNA, pass it through an NGA test and get some data out? This is where the question comes that traditionally a lot of diagnostic tools have used mutations, and mutations are very, very advantageous. They are well approved by FDA for several protocols, but there are only few hundreds in a tumor genome dispersed, need ultra deep sequencing to capture the signal, while methylation can be a few thousand to a million in a tumor. It is spread all over. The signal can be there clustered as blocks. We can capture the entire block. The signals could be as unique to not only detect cancer DNA, but also what tissue it might be possibly coming from. So this is where the advantage of methylation comes. And this brings us to the fact that why these tumor cells would release DNA now. So the release is basically biology, that cell death leads to apoptosis, necrosis, mitosis, there is active secretion, and all of these secretory processes release DNA coiled with histone. This DNA has unique methylation signature, but there is one big challenge here. The challenge is, let's say if I was doing biopsy, and if I took biopsy compared to adjacent normal, I would see biopsy has a majority of tumor cells and a few contamination normal cells, while adjacent normal majority normal cells. In contrast, when I look into blood, the blood of a healthy person and a person, let's say with stage 3 tumor, will still have majority of the signals, which are blood cells, progenitors, vascular endothelial cells, and hepatocytes. The only minute difference would be that 0.1 to 1% of tumor cells, which would be extra coming from the tumor cancer patient. And so we have to design an NGS test, which is sensitive enough to detect that little bit extra and capture those unique signatures. This brings us to an important question, that how much is good enough? And I'll just focus on this middle panel in this plot. So we can all ignore what is there on the left and what is there on the right. What we are looking at is how much should we sequence when we only have 0.1% tumor DNA in our blood? And the answer is, if we just had one site where we want to measure methylation, we have to really sequence at 10,000x or 100,000x to routinely capture that single site, because it's a hit and a miss. This is like a single site. But if we have 10 sites, we can sequence a little less depth and still get the results. If we have 100 sites, even less. And if we have 1,000 sites, we can easily achieve detection of at least one methylation site per patient if we have thousands of those sites. So these kind of simulations give an idea that methylation, even though it's far more widespread than mutation, but still we might need a few thousands of these sites. The range is huge, but at least 1,000 or more to accurately capture with limited sequencing depth, because we can of course sequence to as much depth as possible, but the cost of sequencing also increases. So with all this, we bring forward to you Cancer Spot. This is a study which is the first of its kind. We reached out to different medical centers pan-India. We enrolled subjects. We got their clinical data. We got their smoking history, alcohol drinking history, everything else which is guided by epigenetic, which guides epigenetic signatures. We collected the samples, transported them to the lab, and the samples passed through an experimental protocol which has been diligently curated and refined for it to be functional and successful with very low failure rates. We have sequencing, and the sequencing data is passed through our in-house workflow. That workflow again is highly scalable and again designed to reduce failure rates, any kind of QC failure. And the workflow basically takes cell-free DNA, which is coming out of the blood, extracted from the blood, the methylation signature from the cell-free DNA, going into a machine learning model. And based on the machine learning model, which has been trained on thousands of samples, we finally get a report with a sort of prediction score of the likelihood of cancer being there in the blood of the given individual. This is the number of samples on which we have trained our data, and these are the different cancer types. We've ensured that we try to get as much as early cancer stage patients in our training data so the model learns those early signals. And as we speak, we have collected data for over 5,000 patients across India. Our models are continuously improving, and as we speak, we are working towards next iterations of the model, including even more cancer types, bringing them under the ambit of our cancer spot test. So this brings us to the 5 main factors we have focused on while bringing this test. One is technical variability. We keep working hard. The team puts extreme efforts to ensure that we leave no stones unturned to manage the technical variability. And so we can get similar or consistent signals repeatedly with all kinds of changes in the technical aspects, both experimental and computational. As we saw that tumor signal is scarce in blood, it's 0.1 to 1%. So the DNA fragments which are coming out, cell-free DNA fragments from the tumor with specific methylation signature, they are very small in percentage. So our test has to be designed in a highly sensitive way that we routinely capture those signals, and that we are able to do. We account for methylation variability, conversion efficiency. This is again an experimental challenge to manage that. Whenever we do methyl-seq experiments, the conversion, the artificial conversion of unmethylated Cs to T goes flawlessly, and we have high confidence in it. And finally, lifestyle influence. So we try to keep in our healthy population a substantial fraction of people who are regularly smoking or chewing tobacco, just to ensure that the models don't confuse smoking and chewing as any cancer signal. So our healthy cohorts are not just pristine healthy population, they are also a mix of people who have various habits, but they don't have cancer. This brings us to the accuracy of the test. So currently what we see in our models is we have a 97% specificity. And in stages 1 and 2, we are able to detect over around 10 cancer types with close to 80% sensitivity. This is how the cancer spot report looks like. So there is a cancer spot score associated with the report. And if the score is less than 5%, the signal strength is low. If the score is between 5 to 20%, the signal strength is intermediate, but still risk is not considered high. And if the signal strength is above 20%, we say the risk is high. This is like a positive predictive value. Now, if the signal strength is high, cancer spot not only says that the chances of cancer being in the blood of the given individual is high, it also gives a sort of predicted tissue of origin, the top 2 hits, that we think there is cancer DNA present, and it is most likely going to come from lung or esophagus. Now, as we say, this score, we highly recommend, and we can't stress more on this, that cancer spot is a screening test. And whenever the score is high, even though our models have shown high specificity, decent amount of sensitivity, the recommendation is to please go ahead, schedule a counseling, and please follow up with a clinician for further diagnosis, going through the expected diagnostic route. Now, to conclude my talk, the only things I would like to add is that we've not only looked at cancer signatures in our patient cohorts, we have surveyed hundreds of articles, we have curated thousands of samples from public data, and we have looked into thousands of samples which we have collected. This has led to the various biomarkers we are currently using in our cancer spot test. And one very fascinating observation is that our biomarkers are primarily drawn from an Indian population cohort, that also from the blood of an Indian population cohort. But when we take our biomarkers and test their methylation status in biopsies of Caucasian population, that is the Cancer Genome Atlas data, majority Caucasian, some Hispanic, some Black, African origin data as well there, we see that our signatures derived in the Indian subcontinent tend to also hold in the Cancer Genome Atlas. We regularly see that our markers tend to have a higher methylation score in the cancer patients compared to the adjacent normals. And this also kind of verifies that methylation signatures are pervasive, they are widespread, they span across continents. What we derive here out of blood in an Indian population cohort can very well be seen in Caucasian, African origin, Black population, Hispanic population, all the way in PCGA, which is primarily a biopsy-based database. So finally, to conclude, what we are saying is that methylation is a powerful biomarker, not just in cancer, in multiple diseases. It is predominantly present in blood. We have to target a phenotype, understand its signature, and identify it, and NGS and machine learning makes it possible. And early detection definitely makes a difference. And we have built on these two things to develop our product Cancer Spot, which is clinically available and evolving. And as we speak, we are partnering for progress. And I'd like to thank everyone at NalaGenetics for giving me this opportunity to speak and voice my ideas and facilitate this discussion. Thank you. Just like to acknowledge all the team, and especially Dr. Ramesh and Dr. Vamsi, who are a CEO and CSO, for their continued guidance and mentorship. Thank you.
Thank you so much, Dr. Swaraj, for your very insightful presentation. Okay, so now we're open for Q&A. I think there are several questions here. The first one here is from Dr. Tammy. So she has two questions. The first one, could you discuss the accuracy and limitation of tissue of origin prediction? More importantly, what are the known biological confounders? For example, how do these assays reliably differentiate a true cancer signal from benign conditions like endometriosis or large fibroids that are known to shed DNA as well? And the second question is, could you share some experience on the diagnostic odyssey following a positive high cancer spot score test where the initial workup with standard imaging is negative? So like what's next for the patient? Yeah.
Thanks, Dr. Tammy, for these questions. These are quite relevant. And firstly, talk about tissue of origin. So we do give a cancer signature. And secondly, we give a tissue of origin. That tissue of origin is likely the signature we are getting might be coming from a specific cancer type. Till now, what we have observed is at least one out of the top two, which we predict, they tend to be 77% accurate. So 77% of the times we are correctly able to say in our models and in our test data that test data would be the data which was completely hidden. We never knew anything about it before we built our model. So 77% of the times the correct tissue is identified. Now, confounders, benigns, definitely, as you must have also seen in the slides, we do add a lot of benign population in our data. We deliberately mark the benigns in the control population so that the model doesn't think them as cancers. And we are able to distinguish them because we deliberately put benigns in our control cohort. We let the cancer model understand as so. So, but this is again a continuous work in progress. As we gain more data, as we evolve our patient cohorts and uplift the model, that's where we'll get more improved sensitivity for benigns. Specifically on the diagnostic odyssey, that is very pertinent, very pertinent in these kind of tests, because this is a completely new domain. We are as well learning, and we are very glad to take all the inputs you, the clinicians, the genetic counselors have to provide us how to move forward. What we have noticed is, of course, we can't negate the patient or the person's trauma. That person is nervous while the test is going on, while the reports are coming up. This is not a regular diabetic screening test. This is not a regular blood test. It is something which tells whether we have cancer or not. If there is no imaging is negative, let's say, our recommendation is to follow up with the test in 6 months. Is the signal so early that imaging is not detecting it? So our first recommendation is please follow up with the same blood test in 6 months. If we get a higher signature, we are getting more confidence that in a longitudinal way, progressively, there is some increase in the shedding of the DNA. And second, of course, we recommend to continue discussing with your clinician that based on the tissue of origin suggestions, can some specific scans be done, family history be checked, germline risk be assessed, if not already done. So these are the various channels we are currently guiding.
Okay, thank you so much for the answer, Dr. Suarez. So if I can conclude, there is a 77% positive predictive value, and currently the technology is able to differentiate benign condition, but the model continues to grow. And also for the second question, basically, we would recommend the patient to continue discussing with the clinician, usually following up for the imaging within 6 months.
With the
Okay,
cancer
so
spot test also within 6 months.
okay, with the cancer spot test as well. Okay. Okay. Right. We have another question here from Mr. Santilal Subhash. Were you also
Yeah.
able to identify de novo methylation?
Yeah. Thanks, Andy, for bringing this up. We are working on it as we speak, but our primary approach has been targeted. We want to target a specific set of methylated sites which are present pan-genome. De novo would imply doing large-scale whole genome methyl seq, which is something which was a course we would have followed if our input data was tissue. But with cell-free DNA, to successfully do large-scale whole genome methyl seq, we would need over 100x of coverage, which is huge amount of data and costs involved for that discovery process. I'm not denying that we don't want to go in that direction and understand all the new methylation processes other than what we know very well. But that is something which is a work in progress.
Okay. Thank you so much for the answer, Dr. Swaraj. I think I'll go back to the question that was posed on the first session that I think is good to have now that we have the full picture from your presentation. So from Dr. Taufik, regarding the false negatives, are there known tumor characteristics that are likely missed by the current ctDNA platform, and how should we counsel to patients?
Yes, what we see is that our current models are for early stage, because late stages we have higher accuracy, but late stages there is already clinical symptoms. Sorry, to discuss with the clinician. Early stages, 80% of the cases we are able to detect, but rest of the cases where there is a miss, we did not notice any particular tumor characteristic like a subtype or a histopath or a particular cancer type which is performing less. It's an overall stage-based thing. I currently think it is more to do with shedding. Maybe some tumors like ovarian might shed less than colorectal in early stages. That is something we are looking into. That is something we are working on. But in terms of purely the histopath or the tumor subtype, we haven't seen any clear trends yet. Likely it's the underlying biology, how much the tumor is shedding in the bloodstream, what is coming up, the patient history, the patient lifestyle, diet, so many factors would work. Even their exercise regime would work in these kind of situations.
Okay. Thank you so much, Dr. Swaraj. I hope that answers to Dr. Taufik. Now on to question from Dr. Lona and Dr. Ziske. So what are the limitations on this technology? Yeah, that's the question.
That would be, sorry, from Gabriel.
Oh no, that was from the first session, Dr. Lona Wijaya and Dr. Ziske Maritska.
Okay,
They
from
were
the first
asking
session.
on
Yeah.
what are
Yeah, the limitations is firstly, this is a new technology. Generally, the clinical domain wants something which is highly interpretable. By interpretability, I mean everyone wants to see I have marker A, B, C, D, E. A is present, B is absent, C. As we move towards the AI ML domain, that has become more like a black box for interpretation for a lot of clinicians, counselors, biologists. So they ask how to best interpret the report. We are also working towards it. Of course, we have a huge bunch of methylation signatures, and we see that routinely you take all your signatures, put it in an AI model. The AI model is able to do a far better job than if I would have been staring at each and every report and those thousands of markers by my eye. How AI does it, what is the best guideline to do it, how to get it approved, how to have a standard operating procedure for it. Those are the things I think I see as the major limitation, also philosophically, culturally, how we think of clinical diagnosis and how it is changing. Especially now with LLMs coming up, that is again a whole different ball game, because we are moving not just from machine learning, we are moving into a domain of large language models and transformers, and they look at data in a far different way. So to an extent, I would not be surprised if tomorrow we just sequence something, don't do any analysis, and send the raw sequencing data to an LLM, and that is good enough for it to predict something in future. But this is one aspect I see as a major limitation, that as of now, how to convey best to clinicians how these new technologies are shaping diagnosis.
Thank you so much, Dr. Swaraj. Yes. So, of course, with anything new, it's challenging but also exciting at the same time. Okay, so I hope that answers the question. Now on to Gavian Lua. From a technical or data analysis perspective, how do you determine the methylation conversion step?
So we have our in-house checks in the lab. Thanks, Gavin. You have raised a very critical part of the experimental workflow. We have in-house checks in the lab. We do an early check whether the conversion went well. Once the sequencing is done in mammalian genomes, at least we know that anything which is not a CpG tends to be unmethylated. So anything which is not a CpG in the genome will tend to be converted. And using that metric, we can identify how much is converted well, how much is unconverted. But let's say if the conversion was not optimal, we generally expect more than 99% of conversion in all our samples. We tend to see that in over 95% of our samples. But those 5% where the conversion is not optimal, our algorithm will only take, this is something patented and we have already applied for the patent for it, that we will only take that part of the data out where we have high confidence that the conversion has worked. So we only extract those fragments of cell-free DNA from the NGS data where the conversion has worked very well, and reject everything else where the conversion is not working. So even if the experiment does not look optimal from the lab front, by the time it comes to the computational board, we only extract the most relevant part of it and then only go for the machine learning. So we have these two levels of check, experimental as well as computation.
Thank you so much, Dr. Swaraj, for the explanation. Okay, there is a question from Dr. Raidiana. Do you have an MTA, a material transfer agreement between Indonesia's Ministry of Health, MTO committee, and your lab in India? I think this question is more so for the operations of NalaGenetics
Yeah.
and Strand. So to answer this, yes, we do have an MTA with all of our lab partners, and we definitely follow the guideline and instructions from the Indonesian government, specifically with our health ministry. So uh, yes, the sample will be delivered to Strand Life Sciences in India. Do you have anything to add for this?
Uh, from my side, no, no, thanks.
Okay,
This is entirely your domain. Glad that you.
okay. Thank you, Dr. Swaraj. Okay, do we have more questions? Let me check. Okay, let's wait a few seconds. Anyone wants to ask more questions? We still have time. Maybe, Dr. Zwensi, do you want to add anything else?
Hi, yeah, about I think Dr. Tafik, yeah, about how his questions about how should we counsel high-risk patients who receive a negative MCED result. I mean, it's the same. It's the same with the other result as well. If the patients understand the advantage and limitations of the technology, and we explain everything in the beginning, especially in pre-genetic counseling. That's the crucial part. I think because nothing is like 100% perfect, right? So if a patient understand everything should be goes really well. So the important is we need to open in the beginning about the limitations of each technology that they are using or recommended by clinicians or specialists. That's all for me, Dr. Salma.
Dr. Zenzi, I have a question. Maybe someone in the audience now is very interested to take the test, knowing the technology, understanding all the explanation from you and Dr. Swaraj, but uh, this person is feeling anxious if I will receive a positive result. If you were the genetic counselor, like what would you, how would you, how would you approach this situation? Because the patient actually wants to take the test, but of course, it's normal to have some anxiety for the results.
Yes, because I would too. I would have the anxiety if I'm going to take the same test as well. One thing that someone needs to remember that this test is a screening test, so not diagnostic. And it's something that we can do for DNA methylation specifically. It's something that the main goal is if we found something, we could find it in a very early stage, which is the survival rate would be very high compared if we could find it in later stage on. So this is something that sometimes, no, a lot of time people feel anxious because they do not understand what is this, they do not understand the technology, and and they could process the feeling as well. And patients come to different backgrounds. Some patients with family history, they will come with more anxiety. It's understandable. It's something that we can work on during the counseling. Yeah, you can just call us, and you don't have to do the test. You can just do the pre-counseling and ask questions about all the products in NalaGenetics and so on. And because yeah, I think that's it. Just think about, I mean, anxiety is understandable, but if you could understand what is this, if you get more information, I think it will help a lot. That's it.
Okay, thank you, Dr. Zensi. So knowledge is power. With understanding the technology and what this test is for, the expected outcome and limitations as well, it can give us a more logical reason, yeah, doc, to process our anxiety. And to anyone here, especially maybe Indonesian attendees as well, If you want to discuss more with Dr. Zwensi, you can schedule a pre-test counseling, yeah, doc. So you don't have to necessarily take the test right away, but you can discuss more in detail regarding your own maybe family history and is this test right for you? So feel free to contact our clinic as well. Okay, let me see if there's any other question. I think so far there's no more. Dr. Swaraj, do you have some last things to add?
Nothing specific from my end. Thank you,
Okay.
Dr. Renzi. I couldn't have put it better what you talked about, both the screening and the counseling. So that's very important as we strive to make these products out. It is then the counselors, the clinicians, the patients. There is a huge wide field which is, I would say, I am not the expert in. I am not the expert in how traumatic it can be for people or how people might experience anxiety during these kind of tests. And that's where awareness, the more we become aware, probably the more confident we'll be that these tests are a routine part of our lifestyle, and it's a screening protocol, as Dr.
Yeah. No worries.
Thank you. Thank you so much, Dr. Swaraj and Dr. Zwensi, for both of your sessions that really complement each other. I think we can all agree that we learn a lot during this session. So again, thank you so much for your time. And just a quick reminder to our participants as a prerequisite for getting our certificate, please fill the post-test and training feedback survey. You can scan the barcode here, or you can also check the link that has been sent by our team on the chat box. Okay, next slide. Okay, a reminder again for our special promos. Don't miss them. Whether for yourself or your clinic, now is the best time for you to explore our personalized testing options. So next slide. Yeah, we have our buy one get one free just for today, just for the webinar attendees. We have our pharmacogenomics test that is RX ready that can cover more than 180 drugs. We have Mammo ready and cardiac ready, which are our predictive testing. Mammo ready for breast cancer, cardiac ready for cardiovascular disease. We also have Nutri ready and Life ready. That is for you who want to personalize your nutrition, your vitamin, your supplements, your fitness, skin health, so many things about your lifestyle and wellness in general that you can learn from your DNA and Nutri Ready and Life Ready. And you can also save for with bundles. Next slide. Of course, we have that special 30% discount for the two tests that are on the screen right now, hereditary cancer panel and somatic tumor panel that has been discussed during today's session. So if you are interested, feel free to contact us on the WhatsApp that is found on the screen. Okay. Yes, don't forget to come to our next month genetic essential training series and the next month that we're gonna hold every month until the end of year. So hopefully we can see you again and learn from each other. Before we end, let's take a picture together. Oh, sorry, one more thing. Okay, if you want to get onboarded with us, so maybe you're a physician or your clinic, your facility wants to partner with NalaGenetics, we would love to support you. So we do have an amazing onboarding program where you can learn about genetics. We have a Google Classroom where you can uh, get updated at your own pace to learn about genetics, and you can provide this genetic test to your patients as well. So if you want to get onboarded with us, feel free to contact Ratna. Her number is on the screen, and you will be getting in touch with our team, our medical affairs team, very soon from there. Okay. Lastly, let's take a picture together to document today's webinar. If I can get the audience to turn on the screen, mungkin dokter-dokter sekiranya berkenan boleh menyalakan video, kita sama-sama foto untuk mendokumentasikan webinar hari ini. Okay, let's wait several seconds. I think some videos are turning on now. One moment. Okay, I think we can take a picture now. From the Nala team, would you kindly help? Okay, the co-host already put thumbs up. Take a picture. 1, 2, 3. Okay, I think I'll take my one from my side as well. Okay, 1, 2, 3. Okay, that's all then. Thank you so much, Dr. Zansi, Dr. Swaraj, and everyone here for coming on a beautiful Saturday. I hope it was insightful. And on behalf of NalaGenetics, we really thank you for everyone here, and hopefully we can meet again soon uh, on our next Genetic Essential Training Series. Thank you, everyone. Bye
You.
bye.